Thank you, sir. Thanks for the introduction. Good morning, chairs and delegates. Now, I'm going to present on this managing viral disease of banana to ensure the food security. Actually, before that, I would like to give a brief introduction about our institute. National Research Centre for Banana. It was started during 1993 at Tiruchirappalli, and we are maintaining nearly 540 germplasms of banana. In that, that includes 117 germplasms of exotic lines also, and some wild bananas also we are maintaining. And we have developed some bioreactor technology. Over that, we are multiplying the tissue culture banana plants, and we are commercially supplying to the farmers as well. And we have developed several post-harvest technologies and crop protection technologies as well. So this is the global, now coming to the presentation. Banana in global scenario, banana is a very important crop, not only in the context of domestic consumption and also in the context of export. It is a major crop after rice, wheat and maize, and it is being cultivated in nearly 135 countries with the production of 163 million tons, and nearly 400 million people are feeding this banana. So it is ensuring the food security as well as the for the farmer's point of view, it is giving the very good income generation. And nearly 1/4 of the global production of banana comes from the India, and major consumers are from India and China. And next, when we come to the Indian scenario, the banana total area it is being cultivated in 1 million hectares with the production of 37.47 million tons and productivity of nearly 38 metric tons per hectare. In 1991, it was just around 20 metric tons per hectare, but in 2024 it has increased to nearly it has doubled, 38 metric tons. It was because of the interventions of the tissue culture technology. It has revolutionized the banana industry in India, and major producing states of the India are UP and Maharashtra is the leading producers, followed by Andhra Pradesh and then Tamil Nadu, and other states are also contributing the considerable amount of banana production. When we see the export, it is also being increased fourfold from the year 2018 to 22. It is increasing every year because the farmers they are getting more awareness, and they started exporting to get the more income. So it is a very important export crop as well. And though we are exporting and we are producing so much of banana, so many biotic stress constraints are being faced by the banana growers, which are caused by this fungal disease, bacterial, viral, insect pests as well as nematodes. Among them, viral diseases are very important, and nearly 8 viral diseases are infecting the banana crop globally. Among them, 4 are considered as the major viral diseases in India. That is banana bunchy top virus, then mosaic disease caused by cucumber mosaic virus, banana brack mosaic disease, and banana streak disease. Others are also there, but they are not that much important, and they are not causing the yield loss. First, when we come to the banana bunchy top virus, it is a babu virus. It is transmitted by the aphids. When the virus infects, the plant become bunching appearance, as the name goes, bunchy top. So the top portion of the plant become bunching, and it won't produce any yield. So it has recorded 100% yield loss, and several farms they have stopped cultivating because of this bunchy top disease. They have abandoned the crop. Next, when we come to the banana brack mosaic disease, this is a potty virus. It is also transmitted by aphids, and as the name goes, the brack mosaic, it is causing the mosaic pattern on the bract, which is the spindle-shaped lesions on the bract, and not only on bract, it will produce the spindle-shaped lesions on the leaf, and as well as the pseudostem and peduncle, leaf sheath. As some in extreme condition, it will produce the streaks on the fruits as well, which is later turns into necrotic, which fetches a lower market value. It has recorded the yield loss of 30 to 70% yield loss has been recorded from this. Next one is the banana streak disease. This is a paroretrovirus group. Actually, This is having the nature that it will, during the infection, it will integrate into the genome. There are two different genomes are present in banana, that is A genome and B genome. When they are B genome containing varieties are there, it will, during the infection, it will go and integrate. When the stress comes, then it will again come outside and it will produce as a virus, as we are getting the infection of herpes virus in the humans. The same way it will come. So in a plant, if it is infected, it will produce streaks on the leaves, and later that yellow streaks will turn into dark brown and then black, leading to the drying of the entire leaves. Some banana cultivars are being cultivated only for the leaf purpose. So in that, this is a very major problem. So when the leaf gets infected with this one, they won't fetch the market value. And this is also very important virus in India. Next one is the cucumber mosaic virus. This is not restricted only to cucumber. This is virus infecting more than 1,200 crop species. So it is also transmitted by aphids, and it causes mosaic symptoms on the leaves, and if it is extreme conditions, the entire plant becomes stunting and also causing the hot leaf, which is the youngest leaf, it will become rotting, and the plant won't yield any of the fruits. And these recent days, though it was reported in 1950s, in recent years, for the past 1 or 2 decades, it is emerging as a major problem in the major banana growing areas of Maharashtra and Madhya Pradesh, and in some extent in the Tamil Nadu also. Now, for the managing the viral diseases, I have categorized under the 6 different headings, that is quality planting materials, host plant resistance, biotechnological interventions, chemical methods, biological and cultural methods. In this, first we will see the quality planting materials. So the banana viral diseases are being transmitted through different modes. That is, insects also transmitting, aphids are there, and mealybugs are transmitting the banana streak viruses, and some farm implements during the intercultural operations are also being transmitting. But the major problem is from the planting materials. So this is a vegetatively propagated crop. So when the planting material is infected, it is spreading the virus infection from one field to another field. So the major culprit is the planting materials. Two types of planting materials are being used in the banana. One is suckers and the tissue culture plants. Both are spreading the viruses. So if you see the now the as I told that planting materials are from corms as well as the tissue culture plants. If the corm is infected, single mother plant is infected, it will spread the virus into one plant to 2 plants. If it is through the tissue culture, one plant, mother plant will be multiplied into 1,000, so it will be spread to 1,000 plants. So that much serious it is. As the technology also, tissue culture has revolutionized the productivity. In the equal way, it has increased the virus spread also. So why? Because there are 3 problems are there. Latency is there, virus titer is there, seasonal variation in the symptom expression. Because of this, when we select the mother plant, we could not able to get the virus-free plants. So this make the spread of the viruses. So to address this issue, see, one study was made that in 10 different varieties we have studied. In all the 10 different varieties, the latency was observed, and when the artificially inoculated plants, even up to 450 days of artificially inoculated, they are not producing the symptoms, but the virus are there in the PCR samples. So and even the latent plants, they can be able to transmit. Even 3 to 6% of transmission was also being observed. So to address all these issues, in India, the NCS-TCP has been taken up this tissue culture industry, and who are all producing the tissue culture plants through in India, they should get the certified from this NCS, they should accredit it with this NCS-TCP, and through that they have to follow all the protocols developed by this one. So when they go for tissue culture multiplication, during the mother plant selection, as well as when they distribute the plants infected, secondary hardened plants to the farmers, they have to certify, test the plants for this virus indexing, and it should be certified that they are virus free. Then only they have to distribute the plants. So with this, they can able to reduce the plants. So the virus indexing, they are mainly doing through PCR, ELISA and RT-PCR assay for this BBTV, BSM, MOV through PCR, and CMV and banana bract mosaic virus through ELISA and RT-PCR, they are doing it. So only these two methods are there. Now they have approved for this NCSTCP. Several other methods are also been there for the virus indexing purposes. This virus indexing, I mean the virus diagnostics, it should ensure this this 8 criteria, that is assured. That is the abbreviation. That is assured means A stands for affordable, S for sensitive, specific, and it should be user friendly, and it should be rapid and robust, and it should be equipment free, and it can able to be delivered to the end users. It should be deliverable. So though there are different criteria, different categories of indexing diagnostics are there, it should ensure these things so that we can use it in the NCSTCP. If it is some are highly precise, they are costly, then we can't able to afford for the large extent. So different methods from our laboratory we have developed for the indexing of banana viruses, that is simplex PCR as well as multiplex PCR, and for detecting all the 4 viruses, that is quadruplex PCR also we have been developed in the single assay. And for the quantification, we have developed for the real-time PCR assay, which have identified even from the vector population also. And some extent this banana BRAC mosaic virus we have identified it in the seed, but there is no that much significance of the seed in seed transmission of banana BRAC mosaic virus in banana, but this is also one study we have found out with this. And so from our laboratory we have developed several indexing techniques for which we are following through for the indexing services, which are based on the ELISA as well as the PCR and LAMP RPA based assays. Through this we are regularly we are indexing the private company samples also, and we are earning the revenues as well. And see, with this indexing, so one success story I would like to mention here. Virupakshi is a one banana variety which is widely which is a very good sweetest variety, which is being cultivated only in the lower Palni hills. It is a GI tagged banana, and there they were growing across 18,000 hectares in the hilly area. But due to this introduction of banana bunchita virus in 1970, it has been reduced to 2,000 hectares, because in the hilly area the problem is that the vector population is high. We won't find the vector population in the plain areas, but because of the cool climate the vector population is high in the hilly areas. So because of that, spread was fast, and it has manage the entire crop. So to conserve this Virupakshi variety, that is GI tagged banana, a federation has been formed in 2006 in collaboration with TNAU and our institute to supply the virus free plants to the banana growers. So with that we have developed the mother block of banana virus free banana block in our institute, and through indexing and all, and we have supplied the mother plants to the private companies, and we have supplied it to the farmers. They have supplied to the farmers. So now the area has been increased to 10,000 acres in the Palni hills. So it is through indexing we have able to recover this extinct variety. The same way another I said that banana streak virus, it is also causing a problem in the leaf B genome containing variety. So from our institute, over the 15 years of experiment, we have selected a line which is not endogenously present, I mean which is not integrated into the plant. So that kind of selection we made, and we have identified one line, and it is being circulated to the farmers, and it is giving a very good yield without any virus infection. For past 15 years they are free from virus, that is this banana streak virus. And though there are several advantages are there with the I have mentioned about this tissue culture plants. We can get the uniform growth, not only the virus free, we can get the so many other advantages such as the uniform growth, and they can produce the uniform bunches. Next we will go for the next heading is the host plant resistance. So the host plant resistance, very few germplasms are having the resistance for these viral diseases, that is banana bunchita virus and only BBRMB were identified, but though they identified, they are all not in the commercially cultivated lines, they are in the wild lines. So we could not able to explore, because in banana breeding is very difficult. So only propagation is through vegetative, breeding is very difficult in this, so it may not be explored. So with this disadvantage, biotechnological interventions such as the transgenic development of transgenic banana lines using the hairpin constructs, some attempts were made. By targeting the replicase gene as well as replicase genes for the banana bunchita virus in Rastali variety as well as in the Virupaksha variety, they have developed, and it is working very well. And the same way for banana brack mosaic virus also, targeting the replicase variety, they developed a hairpin construct, and they in the Nagendran another cultivar of banana cultivar, they have done it. From our laboratory also, we have developed the transgenic Virupaksha line with targeting the code protein gene, and the plants are through bioreactor technology we are multiplied, and it is being evaluated. And multiple virus resistance transgenic construct targeting CMV, BBRMV and BBTV were also being developed. And some now the emerging technique, that is CRISPR-Cas9. In this we have targeted the viral genome, because the streak virus they have targeted the viral genome because streak virus is the endogenous and integrated genome. They have targeted it, and they have achieved the resistance for this particular virus. But utilizing this particular biotechnological intervention in the practical point of view, so many biosecurity issues, biosafety issues are there, so it may not be fruitful. So next one is the chemical options, chemical methods. Only this vector, wherever it is present in the hill area, this cool climate, we can go for this managing the vector, but in case of plains it won't work. Managing the vector, it may not be working in the banana as well. So some chemicals, imidacloprid and astaprid, they are using it. In addition to that, some other predators and parasites are also being used for the managing the vectors. One thing, careful thing, since the vectors are hiding inside the crevices of the plants, so they should remove all the trashes, then only they have to go for spraying the particular chemicals. And not only for managing the vectors, we have to destruction of the plants in situ. It is very important, because when we disturb the plant, it may spread the vectors may spread to one place to another place. So we have to destroy the plants in situ by injecting the herbicides. The dosage of herbicide is very important based on the girth of the plant. For every 5 centimeter girth of the plant, we have to inject 1 ml of herbicide that we can able, that is only sufficient to kill the particular plant. And in a single clump, how many plants are there? Each and every plant, they have to be injected accordingly. And next one is the biological methods. Biological methods from our laboratory, we have studied through the Bacillus and Pseudomonas application through root feeding and soil drenching. They have increased the plant vigor, and they have delayed the symptom expression when we apply before the virus infection. Once the virus is infected, after that we are treating, there is no much difference at all. So pre-virus infection, we have to go for this one. And next one is the cultural methods. So several cultural methods I have highlighted, and another one is that including the indexing and vector management. So another one is the fertilizer management also plays a major role in virus disease management. So in case of brack mosaic, from our laboratory study, for the managing the brack mosaic virus, they have increased the dose of recommended dose from 125% and 150%. It has equally, the infected plants has produced the yield, that is 16 kg per bunch has been produced in disease plant as well as a healthy plant by increasing the dose. So, but the same was not reflecting in case of cucumber mosaic virus. When we increase the dose, that same 125 and 150, it is not working here. So, it based on the only 2% increase has been seen here. So, in conclusion, the virus-free planting material is the cornerstone for viral disease management, and preventive approach is very important, and we have to go for an integrated way. It should be farmers should be educated, and in collaboration with the institutions. Thank you.
Long live India and France friendship.